Good day, everyone. For molecular biology and diagnostics, we will discuss your mutation for your period 2. But before we head on to our main discussion, let us just have a quick definition of what is mutation. Mutation is any change or changes in the nucleotide or base sequence of the DNA. It may occur due to errors in DNA replication or due to the impacts of chemicals or radiation to the DNA molecule. So you're very familiar with mutation since it is being discussed during the COVID-19 pandemic. So very familiar na kayo, it is commonly heard yung mutation pagdating sa mga variants of concerns and variants of interest of a certain virus. So mutation can also happen to individuals or persons, even animals that has DNA. So anything na may mga nucleic acid, pwede pong mangyari sa kanila or mutations can happen to them. So, ang mga mutations natin, it can be due to errors in DNA replication, like what I've mentioned during period 1. Yung mga DNA natin, it goes series of replication. And then, during that certain replication, at some certain point in time, nagkakaroon siya ng error. Okay? Minsan, parang random error siya. Paminsan, it can due to external factors, kagaya nga po nung sinabi dito, yung mga chemicals natin. May mga certain chemicals that can alter the DNA and can cause mutation. What else? Yung mga radiation natin, UVA, UVB. What else? Yung mga free radicals natin. Okay, so those factors can affect and alter the DNA and can lead to mutation. Next, mutation may result in coding sequences for new amino acid proteins or not, so it will be discussed later. Next, it is heritable. Yes, it can be herited from your parents, although hindi lahat, hindi 100% mamamana nung offspring ng parents ninyo or kayo as child yung mga mutations na naka-embed sa DNA nila or sa genes nila. Okay? Next, any change in the sequence of your DNA base pairs that is permanent, like what I've mentioned, your mutation can arise by chance, it can be a random error. And lastly, to be considered a mutation, the change in the base pair sequence must not be a result of a recombination. So, to be considered a mutation, dapat hindi po siya recombination. What is recombination? Ito yung exchange ng genetic material of multiple chromosomes kapag kakagaya mo as an offspring, as a child of your parent, na meron kang combination of traits ng parents mo. Okay? Recombination yon. Hindi yun matatawag na mutation. So what are mutations? These are heritable changes in the nucleotide sequence of a given DNA. Substitution, insertion, or deletion of one or more nucleotides. It could affect or not a given phenotype. Phenotype, yung mga characteristics or traits na nakikita natin or parang nagre-result, okay, doon sa mga changes. Okay? So, maaari po na namana mo yung mutation, yung mutated na gene or mutated na DNA. However, it will not manifest. That's the right term. It will not manifest in you. It will not show in the phenotype. Next, major basis of diversity among organisms. It is a raw material of evolution. So, personally, I believe that nothing is permanent. So the only constant thing in the world is change, and that includes a change in the DNA that results to mutation. So it is caused mostly by your mutagenic agents. So what are your mutagens or what are your mutagenic agents? These are substances that cause mutation, either physical or chemical form. And it can be repaired by the body in normal conditions. So may mga genes pa rin naman po na namana natin at some point na nag-mutate siya. However, meron naman pong capacity yung ating body to repair those mutated genes However, kapagka masyado na pong malaki yung nagkaroon ng malaking changes in that DNA or in that gene, doon na siya hindi kakayanin ng katawan to repair. So, magmamanifest na siya, magkakaroon ka na ng different type of mutation which will be discussed in the next slides. So what are the classifications of your mutations? It can be based on nature of occurrence. It can be spontaneous. It arises in the absence of your known mutagen. And it is based also on nature of occurrence due to induced. It is the presence of your mutagens or environmental agents. And it is also can be based on cell type where it occurs. For the somatic, it originates in the mitosis and affects the subset of cells. Sa somatic, mitosis, yung division ng cell mo. And gametic originates in the meiosis and affects all cells of an individual. It can be autosomal and X-linked. So, ayan. Meron lang tayong overview of what are the types of your mutations. It is described in layman's term. So, molecular basis of your mutation. For example, meron kong original na code for your DNA. The cat saw the dog. So, if there's one change in letter, okay, that can be considered as a substitution instead na yung original na nucleotide mo or original na code mo dyan, napalitan ng isang letter for every three and then magko-code siya ng amino acid, it can be considered as a point mutation. Meron ka lang substitution. Another type of your mutation is loss of one letter. It is what you call the deletion. Okay, yung meron mong the cat saw the dog, nabawasan ng isa. Okay, loss of letter C. It is considered as a frame shift mutation. Next, another type of mutation is insertion. Okay, wherein hindi naman talaga originally, wala naman talagang ganung letter. So, nasama siya doon sa tatlong codons na yon. So, nagkaroon ka ng what you call frame shift mutation. So, it will be discussed thoroughly in the next slides. So, number one is the point mutations that affect single sites of your DNA. So, please take note that the point mutations affect single sites on DNA can be a substitution of one base for another, deletion or addition of single base, and then deletion or addition of a small number of bases. For example, yung mga purine mo, okay, or yung adenine and guanine mo, or yung pyrimidine mo, which is your thymine and cytosine, it will substitute for itself. Papalit lang sila. Okay, that is what you call transition substitution. Pero kapag ka naman yung purine mo ay pumalit na dun sa pyrimidine mo, okay, si adenine pumalit na dun sa thymine, okay, si cytosine pumalit na sa guanine, it is what you call transversion substitution. Pag transition substitution, again, kapag nagpalitan lang sa mutation, si A and G, si T and C. So, what are the sample outcomes of your DNA code? So, again, for example, meron kang original dyan. Okay? So, nung template mo, ito siya. Okay? Nung papunta na dun sa mRNA mo, yung messenger RNA mo. Okay? Ito na siya. And then, ito yung mga proteins mo. So, ito yung mga examples ng mga stop codon natin. So, what are your stop codon? So itong central dogma of life. So, from the DNA to RNA to proteins, di ba, meron tayong tinatawag na process from the transcription, translation, and so on. Okay? Pero, forever ba siya na magiging central dogma of life? Ganun yung process. So, hindi po. At some point in time, it has to stop. So, meron tayong tinatawag na parang dulo, na parang tama na, hanggang dito lang. Okay? Meron ka lang na code, nakagawa ka na ng enough protein, and then hanggang dito lang. So that is what you call stop codon. So we have stop codons, and then this is your UGA, UAA, and UAG. Once na na-read na yan doon sa iyong gene or yung nucleotide mo, okay, nag-code na siya. Let's say may nabasa na siyang UGA. O ibig sabihin, magsa-stop na siya sa pag-produce ng protein. So, these are your stop codon. So, hinto na. Hanggang doon na lang. Okay? So, you go away. Yun ang code natin dyan. Mnemonics natin. You are away and you are gone. So, these are your 3 codons na kung saan pagka na-read yan nung iyong template, ibig sabihin, yung pag-synthesize ng protein, it has to stop. Hanggang doon na lang. So, one type of your mutation is the silent mutation. Okay, let us read the description. If a base substitution occurs in the third position of the codon, there is a good chance that a synonymous codon will be generated. Thus, the amino acid sequence encoded by the gene is not changed and the mutation is said to be silent. So, in a shorter term or in a shorter phrase, new codon specifies same amino acid. So let us take a look at the example. So for point mutation, na silent mutation, it has no effect on the protein sequence. So for example, ito yung original mo na sequence. Okay, so yung AGC mo, it codes for serine, valine, protein, and what so on. So nung nag-replicate na siya, okay, pagdating dito sa another template, kung makikita mo yung ikapangatlo, okay, yung ikapangatlong position in the codon, okay, napalitan na siya. Instead of adenine, naging thymine. Okay, however, hindi po nakaapekto ng malaki doon sa amino acid sequence. It still codes for valine. So, wala tayong problema doon. Okay, nagkaroon man ng mutation, very silent, hindi nakaapekto ng malaki. It still codes for the same amino acid, which is your valine. Okay, for example, ito na naman po. Okay, so sa original template mo, due to the redundancy of genetic code, no change in the amino acid sequence is produced. Okay, so that's basically your silent mutation. Another type of your mutation is the missense mutation. This type of mutation is a change in one DNA base pair that results in the substitution of one. It is an amino acid for another in the protein made by a gene. So it means that the new codon specifies different amino acid. So let us take a look at example of your original template. Okay, may AGC, GTA, CCC, and TAC. Okay, nung nag-replicate, meron kang another template. Kung makikita mo dito, okay, AGC, same, GTA. Ano itong codon na to? Okay, napalitan to. Instead na cytosine, naging adenine. Okay, so hindi na po same yung kinode nila na amino acid. So dito, napansin nyo, nagbago na yung amino acid niya. Dito, nagbago na din yung amino acid niya. So that is what you call your missense mutation. Wherein new codon specifies different amino acids. So itong codon na to, okay, nung napalitan, isang letter lang yung nagpalit. Okay, naging ibang amino acid na siya. So that is your missense mutation. Missense mutation produces a change in amino acid sequence in protein product. For example, histidine for arginine may change function or protein or may not. Another type of your mutation is the nonsense mutation. It is also a change in one DNA base pair instead of substituting one amino acid for another. However, the altered DNA sequence prematurely signals the cell to stop building a protein. This type of mutation results in a shortened protein that may function improperly or not at all. So, nung tinatawag nating nonsense mutation is your new codon. Okay, nung nagkaroon ng mutation, nagkaroon ng alteration, ang na-produce niya is stop codon. Okay, so let us take a look at the example below. So, meron ka ditong original template. So, supposedly, meron kang 4 na amino acid. So, nung nagkaroon ng replication in another template, nagkaroon ng change doon sa ika-third na letter. And nung nagkaroon ng change dito sa codon na to, okay, ang pinroduce niya is a stop codon. Okay, so instead na meron ka pa ditong usable or parang significant na amino acid, naging stop codon siya. So, wala na siyang kwenta. Nonsense. Okay, so that is your nonsense mutation. So, for the bad news is that Nonsense mutation produces a stop codon within the a messenger RNA transcript, leading to a truncated protein. How short the protein product depends on where the stop codon was produced within the mRNA transcript. So itong mga codon na to, for example, dito siya nag-mutate, dito nagkaroon ng change. So nagkaroon ng stop codon dito. So instead na meron kang template na mahaba for amino acid, umikli siya, 'di ba? Nawala ng kwenta. Okay, so hindi na siya magpo-function properly. So frame shift mutation, this type of mutation occurs when the addition or loss of DNA bases changes a gene's reading frame. So kaya nga siya frame shift. A reading frame consists of groups of three bases that each code for one amino acid. A frame shift mutation shifts the grouping of these bases and changes the code for amino acid. The resulting protein is usually non-functional. So for example, insertions, deletions, and duplications can all be frame shift mutation. So frame shift mutation is not just one activity happening in a mutation. So it is a combination of different activities such as insertions of a base, deletions of a base, duplications of a base. So for example, insertions or deletions of nucleotides may result in a shift in the reading frame or insertion of a stop codon. So for example, ito po siya. Wait, double check ko yung slide sa susunod. Okay. So going back, frame shift mutation, insertions or deletions of nucleotides may result in a shift in the reading frame or insertion of a stop codon. So for example, yung buong frame na ng template mo. Okay, mayroong nangyaring insertions or deletions. For example, ito. Okay, so nung nagkaroon ng another template, nung nag-replicate siya, hindi na po same yung amino acid na na-produce kasi nga napalitan na yung frame niya. So, how mutations can cause diseases? So, there are pros and cons of your mutation. So, the pros will be discussed in the latter part of this presentation. However, mostly mutations can be can cause certain diseases. For example, yung mga disorders natin such as your cystic fibrosis. Okay, so for the genetic defect or your genotype, meron kang missing na amino acid. Okay, so for the phenotype niya, meron kang lung infection. Okay, meron din tayong, for example, yung Huntington. Okay, ang genetic defect niya for the genotype is that it has an extra base in the gene. Okay, yung gene mo, let's say meron ka lang mga codons doon na let's say 46. This is an example only. So nagkaroon ka na extra na base pair. Okay, so signs and symptoms and the physical traits, the phenotype is that meron kang uncontrollable movement. So yung Huntington disease, yun po siya. So, number 1 kanina is yung, balikan ko yung number 1, point mutation. Okay, that affects single sites on your DNA. Next is the chromosomal mutations change in the structure of your whole chromosomes. Okay, so kung kanina, certain base pairs lang yung pinag-uusapan natin, meron ka lang mga nonsense, missense, mga silent mutation in the nucleotides. This time, yung buong chromosomal mutation na hindi lang po yung template yung pinag-uusapan natin dito, yung buong chromosome mo na. How will I draw chromosome? One moment. Diba? Nagkaroon ka na ng mutation for the whole chromosome. So, marami ka na pong DNA na affected, not just a single site. Okay? Okay. So, chromosomal mutations are more extensive, altering the entire chromosomal structure. These kinds of mutations occur through your deletions, duplications, inversions, and translocations. Okay, so next. Ayan. So, meron na tayong nagaganap dito na deletion of a chromosome. So, number 1 is the deletion of your chromosome. For example, ayan, ito yung chromosome mo. Diba? Ganun yung shape niya. If too much information is lost, it may be fatal to the organism and may result in early death. Example dyan si Cri-du-Chat syndrome. It is a large deletion from chromosome number 5. So, mamaya meron tayong separate slide for that discussion. So, example, ito yung itsura ng chromosome mo, yung isang arm niya. So, nung nagkaroon ng mutation, mas umikli. Okay? Maraming information yung nawala doon sa chromosome na yun. Imagine ilang DNA yung naapektuhan. So, most likely, maapektuhan din po yung function natin as a normal individual. Ayan. For example, meron kang normal hemoglobin. Ito yung kanyang code. Okay, for the mRNA, CTT-GAA, for example. So, nung nagkaroon ka ng mutation in the hemoglobin of your DNA, okay, nag-cause po siya ng sickle cell anemia. So, sickle cell hemoglobin na po siya. Okay, so imagine what mutation can lead to or can cause to a certain individual. So, at sometimes, or pwede po siyang fatal. Ayan. So, these types of mutations, for example, you have a normal gene, nagkaroon ka ng base substitution. Ito siya. Base deletion, instead na may U ka dito, nawala na. So, naapektuhan din yung reading ng amino acid sequence mo. Ayan. So, chromosomal changes can be large or small. However, marami pa rin siyang repercussions in the individual. So, yung chromosome mo, nagkaroon ng deletion instead na may, let's say, meron kang 4 na segment. For example, for that chromosome, may natanggal na isa. So, maraming information yung nawala dyan. Homologous chromosomes, duplication. So, yung dalawa, ayan, nagsama siya sa isang arm nung iyong chromosome. Instead na magkahiwalay sila, homologous po sila. Next is the inversion. Nagpalit yung coding niya, nagpalit yung genes, nagpalit yung possession, and the reciprocal translocation. So, reciprocal translocation is that one part of your chromosome is exchanged with another. Pansin niyo po. Ayan. Okay? It can disrupt functional parts of the genome and have implication for the protein production with your phenotypic consequences. Marami po siyang consequences. So, these chromosomal changes can surely affect that organism. Person man yan, mapa-plant man yan, or mapa-bacteria or virus man po yan. So, duplications within the chromosome. Wait lang na. Babother ako dito, burahin ko lang. Ayun. Okay, so going back, duplications within the chromosome is that if your chromosome is duplicated, the effect of a base duplication depend on the location within the chromosome, whether or not duplication resides in coding or non-coding region of the DNA. So, kung napunta siya sa coding region, hindi sa stop codon, meron din po siyang mga repercussions, kagaya nito. Okay? Next, inversion. Nagpalit na yung mga posisyon ng mga chromosome genes, yung mga certain locations. Reciprocal translocation, yun na nga po, yung sinasabi na one part of the chromosome is exchanged with another. So, it can be caused by due to abnormal synapses event at the meiosis 1 by incorrect chromosomes coming together. So, meiosis nga po yung pinag-uusapan natin dito. It is associated with 2 forms of your leukemia, oncogenes translocated to incorrect regions within chromosomes of your leukocyte or your WBC. So, ito yung mga binabanggit kong mga diseases. These are all genetic diseases. Pero kung i-observe nyo, 'di ba? Kung aaralin mo siya na maigi, galing pala talaga siya sa mutation. Ang background lang natin about leukemia is that it is a cancer of your white blood cell. Pero yung root cause pala niya or yung pinanggalingan pala niya is that it came from a mutation. Ayan, ito yung examples ng mga frame shift natin. And then slippery replication templates. It is a deletion of a base, changes the reading frame of subsequent sequences, and commonly in high repetitive sequences. So, translocation, ito siya. Okay, so example nun yung mga Burkitt's lymphoma, a region from one chromosome, 'di ba nga po homologous yung ating mga ah, chromosomes except dun sa dalawa. So, originally we have 46 in total na chromosome. So, 22 dun, yung 22 magkakapare sila. So, 44 in total. So, yung dalawa hindi kasi yun yung para sa sex chromosomes natin. So, X and X at saka X and Y. So, yung dalawa doon nagpalit. Okay, since homologous sila, nagkaroon ng aberrantly attached to another one chromosome, nagkaroon ng exchange. Okay? Nagkaroon po ng exchange yung chromosome instead na yung code dito, okay, na nandito, napunta po siya dito. So, ganun po siya. So, that is what you call your translocation. So, hindi natin alam na malaki pala yung epekto ng pagpapalit-palit noong ating mga base pairs, noong ating mga codons, kasi magkaroon lang ng slight changes or ng evident changes doon sa iyong nucleotide or yun sa iyong chromosome, it will surely have an effect. Yan. Kagaya nga po niyan, nagkaroon na tayo ng mga leukemia. So, how will we be able to detect mutation? So, for your bacteria and your fungi, we use minimal culture medium, prototropes, and axotropes. So, in the laboratory or in the diagnostics field, we have certain machines that can detect mutations or yung mga resistance markers, for example. So, as we all know, very rampant nga po ang tinatawag natin na antimicrobial resistance. So, these resistant bacteria, di ba nga po, sabi ko, dadating yung araw, pagdating ng 2050, this is coming from a CDC, if I'm not mistaken, dadating yung araw na yung mga bacteria natin, wala na pong makakapagpagaling sa kanila na antibiotic. They are all resistant to the antibiotic. Kasi when was the last time na nagkaroon na naglabas ng new antibiotic? Parang 1950 pa ata. Penicillin pa ata yung huli, if I'm not mistaken. Okay? So wala na silang ginagawang bagong antibiotic. Okay? So yung mga bacteria natin, dadating yung araw na resistant sila sa lahat ng antibiotic, hindi na sila mamamatay doon sa antibiotic. And then what will happen to us? So hindi na tayo gagaling doon sa mga antibiotic na yun. So that resistance of your bacteria are coming from mutation. So galing din po yun sa mutation. So plants, it can be detected through biochemical composition analysis and tissue culture. So going back to bacteria, meron tayong mga machines for that. And then we can use that as medtech in the laboratory sa microbiology. But for molecular biology in the humans, ito yung manner of detection natin of how will you say or paano natin masasabi na meron kang mutated na gene. It is through pedigree analysis, DNA sequencing, and your microarray. So DNA sequencing, it is a very long process kung saan babasahin nung isang machine or nung isang gagawin ng medtech yung process na yon na i-code mo isa-isa yung mga genes. Sobrang ilang merong millions of base pairs na meron tayo sa katawan. So, ayun. Sa cells natin, what I mean. So, yan. Isi-sequence po siya isa-isa and then babasahin yung mga amino acid sequencing natin dyan. So, kung napapansin nyo, yung mga variants of concerns and variants of interest, matagal po siya bago ma-detect. I mean, pwede tayo mag-base sa signs and symptoms. Let's say, si patient A, galing po siyang China. Kung dala na ba niya yung ganitong Omicron B57, ganyan. So, ilang days pa siya bago ma-release kasi medyo tedious yung process on how to do the DNA sequencing. And then, meron lang very limited institutions that does the DNA sequencing. So I think that is in UP or in RITM. So diagnostic tests develop in parallel with particular therapies to ensure the right treatment is given to the right patient at the right time. So we have examples of your FDA-approved drugs and companion diagnostics in the clinical process. So meron tayong approved drug which is the Herceptin. The mechanism of Herceptin is that it targets HER2 to treat the metastatic breast cancer. What is your HER2? HER2 nyo, parang ganun, ginagawa din natin yung mga medtech na mga test, ginagawa rin yan sa laboratory. So HER2 is a protein found on your breast cell. Again, HER2 is a protein found in your breast cell. HER2 stands for Human Epidermal Growth Factor Receptor 2. Again, it is Human Epidermal Growth Factor Receptor 2. So, kung gagawa po kayo ng research sa inyong masteral or sa pagdating nyo ng third year, magandang gawing research is anong magandang alternative, anong magandang component ang tatarget sa HER2. Okay, sa mga metastatic breast cancer. So I just give you an idea. So approved companion diagnostic is that HER2 immunohistochemistry test, HER2 gene amplification test. So we have also the Erbitux and the Gleevec, sorry. Ayan. So ito din yung mga drugs that targets certain proteins in the cells na minsan, kadalasan pala, yung mga cancer cells natin. Ayan. This assay is intended to be used as an aid in selecting melanoma patients whose tumors carry the BRAF B600E mutation for treatment with your vemurafenib. So, mutation is important for evolution. So, hindi naman all the time, puro cons lang yung makikita natin for mutation. If no changes to genomes occur over time, there would be no evolution. So, nabasa ko sa isang book somewhere na nung unang panahon, tayong humans, we can produce our own vitamin C in our body. So, meron tayong capacity to do that. Naka-embed yun sa genes natin. So, over time, nung nagre-replicate na yung mga DNA, hindi ko sure kung ano yung specific reason on why nabura na siya sa DNA natin na nakakapag-synthesize tayo ng sarili nating vitamin C. Pero, imagine, 'di ba? Hindi na natin kailangan kumain ng mga vitamin C rich food, hindi na natin kailangan uminom ng supplements to have enough vitamin C in our body. So, yun. Merong certain point in time na kaya nating mag-synthesize ng own vitamin C in our body. So, yun. And then, sinasabi nga naman na ipagka walang mutation, walang changes, there will be no evolution. Okay? So, meron ding mga magagandang effects naman yung mutation, not just bad. So, meron ding certain point in time na kung saan nakatulong yung mutation para sa mga sinaunang tao na makasurvive sa lamig. Okay? During the Ice Age, I think. So, yun, nakatulong din naman si mutation. So, however, too much change in the DNA is very harmful. And too little does nothing. So, lahat naman ng sobra is mali. A balance exists between amount of new variation and overall health or the adaptiveness of your new variant individual. So, maganda pa rin naman na may mutation. Differences between closely related organisms show closely matched DNA sequences that diverge at some past time and that was adapted for a given environment. So, we still need mutation for us to adapt in the new environment. So, yung mga nag-mutate na kagaya ng mga virus na nakuha natin as an antibody, kinailangan din naman po natin to adapt sa mga paparating pa na panibagong variant of your COVID or your SARS-CoV-2. So our understanding of genes changes with new information is that Gregor Mendel followed traits caused by single gene pairs. T.H. Morgan mapped locations of genes on chromosomes. Jacob and Monod modeled one gene, one polypeptide. Later, it was found some DNA coded for only RNA. Example, your rRNA and tRNA. So I think meron ulit akong nabasa or napanood na it was only 2015 or 2016 kung saan successfully na-code yung DNA sequence in the body. So, sobrang dami. Parang it took them years para ma-code yung genes for a human. So some DNA expresses regulatory genes in some people, and the same DNA may not express in others. So to understand the nature of genes, we must consider their molecular structure, which is the genotype, as well as their phenotypic expression. So genetic code, it is a set of rules which give a relationship between the nitrogenous bases and the amino acid in the polypeptide chain. So we have four features. Number one, the genetic code is the generate. Next, the coding ratio. Pag sinabi natin codon, automatic tatlo yon, tatlong base. Tatlong base agad po siya. Tatlong letter, and then it will code for one amino acid. So, hindi ko na po papakabisado sa inyo yun kasi sobrang dami nun. Sobrang dami. And then there are no punctuation marks to signify the start or the end of the chain. 3 codon will indicate the termination of the chain. That is your UAG, UAA, and UGA. So wag niyo pong kalilimutan yung mga stop codon natin. So the basic principles of your heredity, ayan, pinakita ko lang po yung definition niya, but most likely it will be discussed sa inyong cytogenetics. Pero ang bibigyan ko lang ng emphasis dito is the phenotype and the genotype. Phenotype, the observed character expressed by the gene. Okay, yung mga traits na, yung visible na, yung mga nag-manifest na. Pero yung genotype, set of pattern of genes. Okay, alam na po yung main difference nila. So, autosomal dominant inheritance, ayan, papakita ko lang, included din naman siya. Pero mas bibigang emphasis na po iyan sa inyong cytogenetics. So, summary lang is that most likely 100% or 99% na lang, kadalasan ang mga babae carrier po yan ng mga affected natin na genes. Okay? Bakit? Kasi tayong mga females, meron tayong X and X. Okay, na chromosome. So, kung meron man pong alteration or meron man defect dun sa isang chromosome, at least meron pa tayong isang working gene. Okay, hindi kagaya ng mga lalaki, madaling mag-manifest sa kanila kasi isa lang yung kanilang X chromosome. Okay, so hemophilia, G6PD deficiency, pseudo-hypertrophic muscular dystrophy, the red-green color blindness are examples of our sex-linked recessive inheritance. Wag na wag pong kalilimutan. So, palagi po lalaki yung may color blindness. So, chromosomal abnormalities, number one is the Turner syndrome. Turner syndrome is a chromosomal condition that alters the development in females. So women with this condition tend to be shorter than the average and are usually unable to conceive a child. They are usually infertile because of an absence of an ovarian function. So yun nga po, nagkaroon po ng mga changes sa chromosomes natin. Instead na na-code siya for a certain amino acid, nagkaroon ng alteration. So nagkaroon po ng alteration din sa ating mga organs such as yung mga ovarian function. Kasi itong katawan natin, ano po ito? We are all made up of proteins. Okay, so it alters the development in your females. Other features of this condition that can vary among women who have Turner syndrome include extra skin on the neck. That is what we call the webbed neck, puffiness or swelling of the hands and feet, skeletal abnormalities, heart defects, and kidney problems. So, ito po yung kanilang mga itsura and then yung mga signs and manifestations to them. So, Klinefelter syndrome, also known as your XXY condition, is a term used to describe males who have extra chromosome in most of their cells. Tandaan po, XXY condition. Instead of having the usual X and Y chromosome pattern that most males have, these men have XXY pattern. May extra po silang chromosome. So, even though all men with Klinefelter syndrome have extra X chromosome, not every XXY male has all of those symptoms. Ayan siya. And they have frontal baldness, tendency to grow fewer chest hair, breast development, ayan, female type pubic hair pattern, small testicular size, ayan, kasi nga, nag-aagawan kung female ba siya or male. So, they have extra X chromosome. So, the Down syndrome or the trisomy 21 is a genetic disorder and most common autosomal chromosome abnormality in humans where extra genetic material from chromosome 21 is transferred to a new form embryo. So, kung meron man po kayong pinupusuang asawahin, tignan niyo po yung kanilang genetic background, kung sino man po yung mga kamag-anak nila. Kung meron silang kamag-anak na merong cancer, kung meron man silang diabetes na history, kung meron man po na meron sa kanila na special, parang gano'n. So, yun, medyo mapapaisip-isip ka na. Medyo magiging choosy ka na sa iyong magiging kapartner. So, kidding aside, these extra genes and DNA cause changes in the development of the embryo and fetus, resulting in physical and mental abnormalities. Each patient is unique and there can be great variability in the severity of the symptoms. In patients with Down syndrome, an error occurs in the coming together of chromosome 21. The extra genetic material is responsible for the developmental abnormalities that occur. Again, yung extra na genetic 21 material is responsible for the development ng mga abnormalities. So, instead na 46 kagaya natin, so yung plus meron tayong 2 sex chromosomes, okay, meron silang isang extra. So, ang total nila is meron silang 47 na chromosomes. So, ayan. Ayan po yung itsura nila. Ayan. Ito yung sinasabi natin na cri-du-chat or cri-du-cat, kung ano man pong basahin yan. Cry of the cat syndrome. Ito, rare genetic disorder siya na meron pong naburang portion. Okay? On the short arm of her chromosome, 5 is missing or deleted. Ayan siya. Ayan, high-pitched crying cry of the cat. Kaya po siya tinawag na cri-du-chat syndrome. So that concludes our discussion for your mutation. So for the next discussion, it will be about your chromosomal structure. So if you have questions or clarifications, do not hesitate to message me on my messenger and email during my consultation times. Thank you and God bless.